So hi everyone, my name is John Sanford. I'm a PhD candidate in the Atkinson lab. I work with James, and my talk focuses more on a glycosylation system in syn3, but you'll see some interplaying with stuff that we were just talking about. So the first thing I want to do is just plant some seeds. When we think about a mycoplasma in the natural world, they're chronic symbionts with a host. And philosophically, I would say mycoplasmas are really good at doing two things: stealing things and then hiding when they steal things. And oftentimes you can ask, when an organism becomes this genome streamlined, is why would you make something when you can instead steal it? So with that little bit of background in the way, when we talk about glycosylation in bacteria, oftentimes it's a pretty relatively nascent field still. But a canonical system will use a UDP nucleotide in the cytoplasm to produce a nascent glycopeptide and then export it to the surface. However, the protein hexosylation system I'm about to describe here does not utilize that. Carbon-13 labeling experiments indicate that for or carbon-13 labeled glycoproteins to be found in a mycoplasma, it needs to be the medium needs to be supplemented with C13 disaccharides or larger. So as I basically just said right there, we need a disaccharide or a larger activating sugar to get this process in place. And the other fascinating part about this glycosylation system is it's a very stochastic system. That's fancy English. So more simply put, it's a very random process. There's we haven't found any evidence of a sequence motif like you typically see in a glycosylation system. And instead, it seems to be governed more by the actual biophysical constraints of a protein. So basically, is an amino acid residue available on surface proteins for this hexosyl transferase, an enzyme that we don't know what enzyme it is yet. We think that trying to do homology searches on this will not yield anything of value, because presumably an active site on an enzyme like this utilizing oligosaccharides or disaccharides or polysaccharides is going to be very distinct from a typical glycosyl transferase. The other thing to keep in mind with this system, or the other bit of order to the system, I guess I should rather say, is that once an amino acid residue has been hexosylated by this protein, everything in the immediate area is no longer able to be hexosylated right there. We think this is likely just due to steric hindrance. And when I say immediate peptides, it's probably around 8 or so in either direction of residues. But it's kind of, once again, governed more by how the 3D space is available to the protein. Also of note, we see that a pretty wide range of residues able to be hexosylated, including asparagine and glutamine. That one is incredibly atypical for glycosylation systems, as well as serine, threonine, and tyrosine, which are relatively more common. Tyrosine's a little bit of an uncommon one, but it's not that unheard of. So SDS, or when we search for glycosylation in mycoplasmas, we typically utilize a paired SDS page approach, where we run two gels with the same quality and quantity of material, stain one with Kumasi or whatever protein stain you want, and then another one using ProQ Emerald 300. It basically uses periodic acid to allow fluorescence of liberated ketones and aldehydes. And synth is an annoying little organism when you try to run isolated surface proteins through an SDS page because of that beta-galactofuranose capsule on it. So my good friend Jason Needham provided me a script to show areas of hyper and hypoglycosylation, or in this case, more accurately, this is relative pixel intensity between the Kumasi region and the analogous region of that glycostain gel. So here to here, for example, I'm sure your eyes can make this out. You can see that there are areas where that glycostain jumps out from this background, but it's also denoted by these more red and yellow regions here on the figure that I've generated here. So we found evidence of glycosylation in this organism. We see this in other mycoplasmas. In fact, we've yet to find a mycoplasma that seems to have a complete glycosylation knockout. So in the context of this talk, we proceeded to move forward to do glycoproteomic analysis. And so a workflow for this is we culture the organisms and harvest them. After lysing the cells, I have M. genitalium and syn3 here for examples. We ultracentrifuge the membranes after lysis, perform an in-solution trypsin digest on these centrifuged membranes, and then do a chloroform methanol extraction, because in our experience, trying to run syn3 membranes on HPLCs and whatnot is a very messy endeavor and often has solubility issues. So by chloroform methanol extracting the digested peptides, we load in both the aqueous and the organic phases, leaving behind the interphase that causes a lot of the solubility issues. And most of the data in fact, all the data I'll be showing going forward here is through Orbitrap mass spectrometry. That's a high resolution technique. However, we're getting ready to do ultra high resolution Fourier transform ion cyclotron resonance mass spec, which is just essentially an incredibly high resolution MS technique. So then we do our data analysis, which will eventually, the mass spec fairy lets me get identified glycoproteins that way. Using this workflow, we found some putative hexosylated proteins through this Orbitrap mass spec. I, of course, say putative because we still need to go through and verify some of these. However, from the preliminary analysis we've done, they're passing a lot of statistical checks and whatnot. So I'm feeling decently confident that these proteins have been identified as hexosylated surface proteins. And we'll show you some more information about that going forward. So this first system right here is an O-linked hexosylated peptide from a one that's annotated as a fatty acid binding protein. Here's the accession number. And this tyrosine residue right here has been hexosylated on this particular peptide. We note that by the 162.02 Dalton shift, or 0.05, sorry, need to finish my coffee. But with that corresponding mass shift, that's an indication of a hexose being attached to it. That could be a wide range of different hexoses like glucose, mannose, or galactose most likely, but we're doing more stuff to confirm that. And by comparison, I should say when we get these generated data sets, a very small minority of those peptides are actually going to be glycosylated. Like this is probably or if I had like 100 copies of this particular peptide right here, TYN, onwards, only about probably like 5 or even less of those peptides would have this hexose on this, maybe once again, even less than that. So we have a much more typical or much more abundant unmodified equivalent peptide in our region, or in the same, what's the word I'm looking for? In the same spectrum, I should say. And the unmodified one has a much shorter mass on the tyrosine residue, specifically 162 or minus 162 Daltons, aka the unmodified peptide. So through these comparisons right there, we see evidence of that stochasticity I was just talking about earlier. Likewise, we found evidence of N-linked ones. I will probably stick this in a supplement on the upcoming paper I'm working on, because we got some pretty lousy fragmentation on this latter part of this peptide sequence, but we did get evidence of a hexosylated Q residue right here, or glutamine, I should say, which once again is a very atypical amino acid to get hexosylated. So, and likewise, I got a much more abundant version of this N-linked, or what is in this case N-linked hexosylated peptide right here. If I forgot to say, this is the N-acetyl mannose-amine 6-phosphate 2 epimerase protein. So we're continuing to go through these and find these modified peptides. Our kind of holy grail that we're looking for is getting an unmodified peptide and then 2 of the same peptide length with different glycans attached to different areas to show the stochasticity, but we're still working through on getting that in syn3. So the conclusion from this is that we have protein hexosylation maintained in SYN3, despite the synthetic genome reduction. In other mycoplasmas, as I mentioned earlier, it's usually either mannose, galactose, or glucose, which it's scavenging from the growth medium. And the part that really intrigues me about this, especially with synth, is this hexosylation system appears to be either essential or quasi-essential. I still need to go back and try to run this with SYN3A in the future. And that leads to an interesting question on what it's doing. The first thing that pops into mind, a stochastic surface protein hexosylation system would I'd be more than willing to bet money is increasing antigenic variation, but that just seems to be a secondary role. Perhaps it's protecting the host cell from the proteases that mycoplasma secrete. Once again, why make something when you can just steal it? Lastly, in the future, I'm currently performing gas chromatography mass spec on SYN3 that's been grown with SP4 that's been spiked with different disaccharides to see if I can manipulate these ratios of hexoses relative to the control. I'll get back to you on that in the future. Next month, I'm going down to the highest resolution mass spectrometer on the planet, actually, and running SYN3A surfaceome stuff, or I say getting some evidence of the SYN3A surfaceome or getting a feel for it. Anytime I run things on this, the raw data will be uploaded to the repositories when we publish it. So if you're interested in ultra high resolution surface omics and synth, get in touch with me or look for the repositories when they go up. And then in the future, we're doing more surface omics and moonlighting stuff. With that, I'd like to thank my lab and collaborators, and I'll be happy to take any questions you have.
John, it would seem like there should be sites in proteins that you suspect of moonlighting, that there would be amino acids we couldn't mutate. So that, for instance, AlphaFold may suggest that you could replace a serine with something else at this site, but by losing the glycosylation site, we would either make a non-viable cell or ideally a cell with a different phenotype. If you guys could make us a list, not too big a list, of some mutations you would like us to build you, this could be done.
That sounds great. I think that would be more relevant for looking at this rhamnosilation system that James mentioned, because we believe that this one is more of like an artifact of, or not artifact, but more of like a like an effect of once a protein has come onto the surface, whatever hexosyl transferase is doing this process is sending or stochastically hexosylating things on the surface. And as I said, we speculate it's just to protect this, its own surfaceome from being degraded. So I think it's not so much of like mutating things, as long as that amino acid is available to be hexosylated, it wouldn't matter if the mutation, say changed from like a threonine to a serine or something like that. Once again, so long as the 3D space is available.
Okay.
But like I said, I think that's a really good idea for looking at the moonlighting process. So I definitely appreciate that.
Other questions? I'm expecting to hear something from the people in Illinois if they haven't had to go to class or something.
Hi, John. James Pelletier here. Beautiful talk. I'm curious, some of the moonlighting proteins that might not have signal peptides, do you have a sense how they're exported to the surface of the cell?
I think, once again, jumping back to James's talk, that one of the hypotheses was that rhamnosilation system is what actually signals or allows a peptide or protein to be sent to the surface. This one, once again, is more of a consequence of once it ends up on the surface, it's viable to be hexosylated. But thank you. That's a great question.
Thank you. So then a follow-up question: if it's the post-translational rhamnosylation, is the thought that they'd be exported in either a folded or partially folded state?
That's a good question. I would presume that once the rhamnosylation has taken place on it, that it either is folded in a way that allows that export to take place, or as James said, you can either prevent, like, say, oligomerization from happening with other parts of a or protein complex.
Cool. Thank you so much. Beautiful.
Thank you.